Welcome to your online lab for week 4. So it's our second online lab. Now, what are we going to talk about today? Well, as usual, we're going to address learning outcomes that pertain to the topic at hand, which is going to be osmosis diffusion. We're going to take a look at some Punnett squares and tonicity as well. And we're going to do some hands-on activities, but I'm going to be the one doing the hands-on activities. However, you might be able to do some of these experiments at home. What learning outcomes are we going to address? Well, to be specific, we're going to outline the physiological significance of electrolytes and how they contribute to pH, so some pH experiments. We're going to focus on diffusion and osmosis, like I stated, and then try and apply our understanding of diffusion and osmosis when it comes to tonicity, which is very important medically. And then we're going to take a look at membrane transport and so forth, plus Punnett squares, which I didn't put up here. The experiments I'm going to show you today is an osmosis experiment, a diffusion experiment, a pH experiment, and then we're going to go through those Punnett square questions right at the end. So the first experiment or activity I want to do is I've boiled 2 eggs. And we're going to place those 2 boiled eggs inside 2 containers. One container contains 0% salt or sodium chloride. The other contains around about 20 to 30% sodium chloride or salt. We're going to weigh those boiled eggs before we chuck them in, and then we're going to weigh those boiled eggs after and see what happens. So let's go and do this experiment, shall we? All right, so what we need for this experiment is 2 boiled eggs, we need a weigh station, and we need 2 bowls. One bowl will contain 0% sodium chloride, so pure water, and the other bowl contains 30% sodium chloride or salt. Now the first thing we need to do is take these 2 boiled eggs and we need to weigh them individually and write down what their mass actually is so that we can compare it to after the fact. So let's take the first egg, turn the weigh station on and place the egg. And you see that this egg weighs 52 grams. So we can write for the 0% sodium chloride, this egg weighs 52 grams. Now we can take this egg off. We can grab the second egg. Well, first let's place this egg in the pure water in the 0% sodium chloride and we'll wait. Now let's take that second egg, let's weigh it. And you can see it weighs 51 grams. So for the 30% sodium chloride, before we chuck it in, it weighed 51 grams. So now let's take this egg and chuck it into the 30% sodium chloride. Now all we need to do is pretty much sit back and wait and see what happens. All right, we're back. Here are our eggs. 2 hours have passed. We need to turn the weigh station on first, and then we need to take the eggs and weigh them and see if there's been any weight changes. So first egg, let's take this one out. This was the 0% sodium chloride, so just pure tap water. It weighed 52 grams before we chucked it in. And now let's weigh what it is after being in for 2 hours. We'll dry it off first very quickly. Get all the excess water mass off, and now let's chuck it on. It's 55 grams. It was 52 grams before we chucked it in, now it's 55. So its post weight after 2 hours, being in pure water, is 55 grams. So what this means is it's gained 3 grams of mass. So 3 grams of water mass. So now let's take it off. We'll chuck it back in, just leave it there for a little bit. Let's now take the egg that's been in the 30% sodium chloride. Beforehand it weighed 51 grams, so let's dry it off a little bit and chuck it on the weigh scale and see what we've got. It's 51 grams, so it's hasn't really lost any mass, maybe a tiny bit that this weigh measure can't detect. Probably half a gram or so it may have lost. So we'll we can say that its loss is 0 grams, but let's just assume that it lost 500 grams because that's probably more accurate. But what we've highlighted is that the 0% sodium chloride has gained some water mass, and in the 30% sodium chloride, it's lost a little bit of water mass. So why would we expect to see a boiled egg? So let's draw it up actually. Let's have a boiled egg, and we place this boiled egg into a container, and we know we've got 2 boiled eggs, right? So let's place both boiled eggs into a container, and this container is filled with water, except what we know is that this one has 0% sodium chloride, so salt, and this one has 30% sodium chloride, or salt. Now remember, what we saw was that this egg tended to gain weight. It gained mass. It gained mass. Now this one we're expecting mass to be lost, but it didn't. But let's just say it did, all right? Let's, for this thought experiment, that it loss of mass. Now the question is, why? Well, think about it. If you've got an egg, that egg in this scenario is basically a cell. So it's got a semi-permeable membrane on the outside. And you know that inside the egg there's going to be a certain concentration of things, ions, for example, sodium chloride, potassium, magnesium, and so forth. There's going to be a certain concentration of those things inside the egg. So let's draw up 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 particles or solutes. Remember, a solute is a substance that gets dissolved in water. So let's say there's 10 solutes inside this egg, and same here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in this scenario, this egg with its solutes are placed inside of this water that contains 0 solutes. There's no sodium chloride out here. So what we've got is a concentration difference from inside the egg to outside the egg. And remember, the egg is a semi-permeable membrane. It only lets certain things in and out. It doesn't let ions in and out, so sodium and chloride, because they're charged. Remember, if it's large or charged, it can't go across the cell membrane. So remember, don't ever forget, when there's a membrane separating out 2 areas, the whole aim is that it wants the inside to be, or one side to be the same concentration as the other side. And it can do it one of 2 ways. It can either let those particles or solutes diffuse out until they're the same concentration, or if that's not possible, you can change where the water moves to change the concentration. So in this case, it's a higher concentration inside the egg compared to outside the egg. Now, the term we use here is that the solution it's bathed in is a hypotonic solution. Hypo means below. Tonic is concentration difference. So the fluid here is below the concentration of the egg. Let's go over to this scenario. So now the egg is placed in a 30% sodium chloride. So the solution that the egg is placed in is way more concentrated than that of the egg. Let's assume that the egg is 0.9 sodium chloride like we are. So you've now placed the egg in a solution that has way more salt. So there's a concentration difference, but this time it's the solution that the egg is in that's more highly concentrated. So we call this solution a hypertonic solution. Hyper means greater than. So the solution has a greater concentration than that of the egg. Now again, we want the concentration inside the egg to be the same as the concentration outside the egg, or at least it wants to try its hardest for that to happen. In this scenario, it would want the solutes to go out, but it can't. In this scenario, it wants the solutes to go in, but it can't because, again, the egg has a semi-permeable membrane. So what actually happens? Well, water moves. Water can freely move across this membrane. And so in this case, to balance out the concentration, water will want to shift into the egg. Now think about it like this. Think about there's a tug of war happening from one side of a membrane to another. Whichever side has the most solutes is going to have the strongest pulling force. So it wins the tug of war. So in this case, the egg is pulling the water inside. And what happens to the egg? The egg begins to swell. So when we took the egg out and weighed it, it gained mass because water moved inside to try and balance out the concentration difference. In this scenario, there's a higher concentration of stuff outside. So we want to balance it out. So the water will move out to try and dilute that concentration. So the water moves out to dilute the concentration to try and make it more similar. So that means it loses, it shrinks, right? The term we actually use is crenate. It will crenate. That's another word for shrink. So that when we take it out and measure it, what happens? It loses mass. So this is actually the application of osmosis in the context of tonicity. So now let's take a look at the diffusion activity. So we're going to take a jug with water and we're going to put some food dye in it and measure the rate of diffusion of the food dye. So this is an experiment you can do at home. Seems very simple, but it highlights different factors that can affect diffusion for us. So the way we're going to do this is we're going to take some red food dye and a jug of water. So make sure the volume of water you use every time is the same. And we're going to take 1/2 ml of red dye. So hopefully I can show you that. There we go. There's 1/2 ml of red food dye. And we're going to squirt it in and we're going to time how long it takes for it to diffuse relatively evenly throughout this cold water. So let's take a look and go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, it's going relatively slow here. So you can see that we're at around about 14, 15, 16. It's probably diffused halfway through this water. Let's look at hot water now. So we've got our hot water here. This is definitely hotter than what we just had. Take 1/2 ml out of the syringe. It's still 1 liter of water, 1 cup, whatever it was I used. And we take that 1/2 ml, and now let's see how long it takes in the hot water. Ready and go. 1, 2, 3, 4, 5, 6, 7, 8. Look at that. It's 8 seconds. It's already diffused all the way through because of the hot water. Why? So what happened in the scenario where we had 2 containers? We put that red food dye, remember? So we put the same quantity of red food dye in. I think it was 1/2 ml we put into each. So that was identical. So that didn't change. The volume of fluid was identical. So the volume of fluid was both, I think it ended up being 1 cup of water. 1 cup of water. So those factors are identical. The only thing that was different was that this was cold water and this was hot water. And what happened was we put the food dye in, the red food dye in, and we watched the rate of diffusion. So remember what diffusion is. Diffusion is when you have a high concentration of particles or solutes, that it will go from its area of high concentration to its area of low concentration to try and evenly distribute its particles to create an even concentration. The same thing happened here. It happens all the time when you put a tea bag in water, for example, diffusion occurs. But what we saw was diffusion in the cold scenario happened more slowly than the diffusion in the hot water. This happened more quickly. And so the question is, if I learn how to spell quickly, the question is why? Well, it all has to do with factors that affect diffusion. When water is cold, the medium, the solution of water, so you've got the solutes, which is the red dye, and the solvent, which is the water. When water's cold, or any particle's cold, it's moving very slowly. When you add temperature to a particle, it starts to move super quick and it's bouncing off, it's moving around, it's bouncing off the walls, it's moving really, really quickly. Which means when you put this solution, or I should, the solutes into the solvent here and it's hot, everything's jiggling around really quickly. which is basically just like you shaking it up. So you're adding energy into the system. Adding energy means it diffuses far more quickly. Here, because it's cold, there's less energy. Things are moving more slowly, and so it takes a lot longer for those particles to diffuse. So that means that one of the factors that affects diffusion is temperature. Temperature can affect diffusion. What's another factor that can affect diffusion? Well, the size of these particles. Size affects, now you might be thinking, how? Well, think about it like this. If you've got a cornfield, right, and you had a mouse run through the cornfield, it could run through super quickly because the corn stalks are not in the road for that mouse. It can just move through very quick. But if you've got something a lot bigger that needs to run through, like a dog, for example, it's going to get caught up. It's going to hit the corn stalks and it's going to be moving through more slowly. The same thing happens for diffusion, depending on the medium. So for example, if you were to have an agar plate, right? It's agar. And you were to put two different chemicals on that agar plate. Let's draw it up. This is what's happening in the labs this week. You've got an agar plate and you put a chemical here and a chemical here. But this one has a molecular mass of 300 and this one has a molecular mass, so this is just its weight, right, its size, of 50, and you watch the way it diffuses through. This one's going to diffuse more quickly because it's smaller. It can move through the medium far more quickly. This one moves more slowly because it's very big. It's hard to get through. So size. So we've got temperature, we've got size. What if we were to add external energy? So we could add kinetic energy to it. What am I referring to here? Kinetic energy is if I were to get a stirrer and put a stirrer in and start doing this, diffusion rate's going to increase super quick because again, I'm adding energy into the system. That's all the stirring is doing. So there's a couple of different factors that obviously can affect the rate of diffusion. And the reason why we're going to teach you this is because of drugs. All right? So the size of a drug can affect its diffusion rate, as an example. All right? The medium in which it's moving through can affect the diffusion rate, and so forth. So quick 101 on diffusion. So in this next experiment, we're looking at the role that pH plays biologically in our body. So what can pH do to affect the structures of our body by specifically looking at proteins? And we're using eggs as an example. So what we're doing is we're cracking 2 eggs, one into pure water, which we can see here. Water has a pH of 7. And so there's the egg cracked in tap water. It's been in there for around about an hour. And then over here we've got another egg cracked, but this time in vinegar, which we know has a pH of around about 2 or 3. And again, that's also been in there for an hour. Look at the differences. This egg in the water, you can see still quite translucent for the egg white, but this egg white started to look foggy. Starts to look quite white. It almost looks as though it's starting to get cooked. I reckon if I left this egg in the vinegar for long enough, it would actually look as though it's entirely cooked. So the differences are quite pronounced, and this is only after 1 hour. So why are these differences present from pH? Okay, so why, when we crack an egg into normal water, so this is just tap water, which we know has a pH of 7, compared to when we crack an egg into vinegar, which we know has a pH of around about 2, how come the white of this egg looks very, not transparent like it does here or translucent, but actually quite white, almost as though it has the appearance of it being cooked? Why is that? What's happening in this scenario? Well, we need to go back to the fact that eggs contain huge amounts of proteins, which then means we need to go back and talk about what we spoke about a little while ago in regards to what proteins are. And remember that proteins are just these big long chains of amino acids that fold in upon themselves. So remember, these amino acids have different side groups, which I said are like personalities. So for example, I'll get another pen. You can have an amino acid. So I'll just write it like an amino acid, an amino acid, an amino acid, an amino acid, an amino acid. Remember, they're all connected in this big long string of amino acids, and they've all got different side chains. So one side chain may be hydrophilic, so it loves water. Another one might be acidic. Another one might be basic. Another one might be hydrophobic. Right? And let's just say this one's acidic as well. So remember that when they spontaneously fold to turn into that three-dimensional protein, because proteins fold and they have surfaces and areas where things can bind, or maybe they act to carry things away, or they can work as receptors, it's all because of the way it folds. And it folds in accordance with these side chains. And it folds because the solution that it's in is pH 7.35 to pH 7.45. That's the narrow range that it adequately or appropriately folds in. So for example, this amino acid is basic at this pH. This amino acid is acidic at this pH. But if you start to, and same with this one, and so it folds a particular way. But if you put it into a pH of 2, you actually start to change these side chains. And the acidic basic ones, they don't fold properly. Instead of this maybe folding inside, because the pH is totally different, it opens up. And so what you end up getting is proteins going from its three-dimensional folded shape to a more open and linear shape, because it can't fold right. Its folding is entirely in accordance with this pH range. But if you change the pH range, well, what happens? It opens up. It can't fold properly. And so this process is called denaturation. Denaturation. Now, your question might be, why does it look as though it's cooked? The egg looks pretty much similar to the way it does when you put it into a frying pan, because the folding of proteins also happens at a particular, not just particular pH, but a particular temperature, which is around about 37 degrees. So if you heat a pan up and crack an egg in to, let's just say, 100 degrees, the same thing happens. These amino acids and side chains, they fold in accordance with a good pH and a good temperature, right? Which is that. You knock any of them out of whack, it unfolds the protein, and that's called denaturation. And it actually makes it easier to ingest, to eat. Why? Because then little enzymes can come along and chop up the protein into smaller little pieces for us to absorb. This is why we cook meat, because cooking it denatures the proteins and the enzymes in our body can chop it up. Then we can chop off these amino acids super easily, absorb them individually, and utilize them elsewhere. So in this experiment, we've got a couple of test tubes with different types of acids, bases, waters, and so forth. And it's your job to test the pH and then explain why is that pH the way that it is. So we've got one test tube that's 100% acid, one that's 100% base, one that's 50-50 acid-base, one that's 50-50 acid with water, and one that's 50-50 base with water. You then get your pH meter and you measure the pH, and what you find is this. The pH of the 100% acid is a pH of 1. The pH of 100% base is a pH of 14. The pH of the 50-50 acid base is a pH of 7. And the pH of 50-50 acid water is a pH of 1 to 2. And the pH of base to water is a pH of 13 to 14. So you're looking at this and you're thinking, well, okay, pH of 1 with an acid. What's going on here? Well, we know that an acid, when we measure an acid and the pH is really low, we're simply measuring. Remember, pH, the H is measuring the concentration of hydrogen ions. pH equals what we call the negative log of hydrogen ions. We're measuring the concentration of hydrogen ions, that's it. So if something's a pure acid, 100% acid, it means that we basically have a whole bunch of hydrogen ions in this solution. That's it, free hydrogen ions. If we measure 100% base, It's basically saying we're measuring the fact that there's no hydrogen ions in here. In actual fact, we're measuring something. Well, we're not measuring, there's no hydrogen ions. Remember, pH is only the measurement of hydrogen ions. If it's 14, it means there's really, really very, very few. And the reason why is there's something in there that seems to be mopping it up, grabbing it and holding onto it so you can't measure it. Something like hydroxyl ion. Because remember, the definition of an acid is it donates, it's any solution that donates hydrogen ions. That's an acid. So it releases free hydrogen ions. And then when you measure the pH, you're measuring how many of those free hydrogen ions there are. The definition of a base is that it accepts hydrogen ions, which means it mops it up and holds onto it. So when you measure it, the pH meter goes, well, no hydrogen ions here, you've got a basic solution. All right. So an OH is a really good chemical to bind to hydrogen ions. So pH of 1, a pH of 14. Again, I just want to highlight this to you. If you look at the pH scale and we go 14, 13, 12, 11, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, right? That's the pH scale. 7 is neutral pH. Now what this means, because I said it's the negative log of hydrogen ions, and you're probably thinking, what the hell is that? This simply means that there's 10 to the negative 7 hydrogen ions in the form of molar. And you're going, what are you talking about? Have a look. If I write 1.0 and the concentration is molar, and you're probably thinking, I don't understand that concentration. Don't worry, you'll do that in medications. So you've got 1 molar. If you move to negative 7 decimal places, 1, 2, 3, 4, 5, 6, 7, that now means, if I put the zeros under here, the concentration, the amount of hydrogen ions that we have in this solution is 0.000001 molar. That's the concentration of hydrogen ions in a pH 7. So that's not many hydrogen ions. If we go down to a pH of 1, it simply means it's 10 to the negative 1, which means if you have 1.0, you move it back here. And so now the pH is 0.1 molar of hydrogen ions. Have a look. This number, this concentration is way higher than that, right? You can actually have a pH of 0, and that simply means it's going to be 1 molar of hydrogen ions, okay? So this is what we're referring to. So if you go to a pH of 14, simply 1.0 and go back 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 decimal places. So it is a really, 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 really small number. That's the concentration of hydrogen ions in a pH 14. Hence why I told you that a base has no, basically none, very small amounts of hydrogen ion, because OH binds to it. Now I can demonstrate this nicely to you here when we evenly mix an acid, hydrogen ions, with a base, hydroxyl ions. What do we get? Well, if I told you that OH loves to bind to hydrogen ions, so I said OH negative loves to bind to hydrogen ions, what do you get in this beautiful relationship? 2 hydrogen, H2, the negative and the positive balance each other out, and then an oxygen. You get water. You get water. So what that means is, and what did I say? Water is a pH of 7. Now this starts to make sense. You've got a pH of 1 here for the hydrogen, a pH of 14 here for the hydroxyl. You bring them together, they balance each other out, and you've got a pH of 7, which is the pH of water. So that's why it has a pH of 7. Hopefully that's starting to make sense. But if you have acid mixed in with water, well, the hydroxyls that are present in water are already bound up with hydrogen, which means that the hydrogen that's now free in this solution can't bind to anything. So you still have the same number of free hydrogen ions. It's just slightly more diluted in a larger volume, but the number of free hydrogen ions are still there. So the pH is still going to be very similar because you've still got free hydrogen ions. That's all you're measuring. So if you wanted to go from a pH of 1 to a pH of 2, so that's 10 to the negative 2, which is going to be 0.001 molar. So it's 10 times the concentration difference. So if I wanted to go from a pH 1 to a pH 2, I would need to put 10 times more water in this solution than I have hydrogen ions. 10 times. So if that's 1 litre in that, let's just say that's 1 mil in that tube, I need to put 10 mils of water in that tube just to go from a pH 1 to a pH 2. But with the acid base, if I have 1 mil of hydrogen ions, if I add simply just 1 mil of base hydroxyl ions, it's going to go from 1 to 7, right? So that's the point I'm getting across. And the same thing's happening here with the base. When you put hydroxyl in and you put water in, again, nothing's happening. Any hydrogen ions that do get released from the water are going to be bound up, I should say, by hydroxyl. So the pH barely changes. Hopefully, this helps to make sense of this experiment. Let's now take a look at Punnett squares. So the first question you have is, if both parents are carriers or heterozygous for a recessively inherited gene or disease, what percentage of offspring will also be carriers for this particular disease? Let's take a look. So there's a couple of terms here you must understand. Firstly, remember that we have 2 copies of every gene. Remember, we have 23 pairs of chromosomes. They're pairs. So I get chromosome 1 all the way to 23 from my dad, and I get a copy of chromosome 123 from my mum, and they come together. So that means I have 2 copies of every one, which means all the genes that can be expressed into proteins, I've got 2 copies of. Generally speaking, the version I get from mum and the version I get from dad are identical, and that's fine. So if they're identical, you can just denote, let's just say the gene, let's say in this scenario, gene is going to look like an A, right? So I can get my 2 copies, right? I can get 1 copy from my dad, which may look like that, and 1 copy from my mum, which may look like that, and they're identical. If that's the case, I'm termed homozygous. So homo meaning same. Maybe the genes can be denoted like this, which it's just saying that, oh, this gene is a little bit different. And maybe I get that version from dad and that version from mum. So they're slightly different. The term we use here is heterozygous. So hetero meaning different. And the question asks about the fact that 2 parents are heterozygous, and the term we use, we also can say carriers. So let's just say the dad, he has a big A and a little a, and the mum has a big A and little a. They're both heterozygous, they're both carriers. And the question asks what percentage of offspring are also going to be carriers. Now you could say that this little version here is the version where you don't get the disease. This one, however, is the version where you do. But they're carriers. Because it's a recessively inherited disease, it means you need 2 copies of the dodgy one to get the disease. If it's dominantly inherited, it means only 1 copy is necessary for you to get the disease. I'm going to say it again. Recessive inheritance means you must have 2 dodgy copies to get the disease. Dominant inheritance means you only need 1 dodgy copy to get the disease. So here, mum has 1 dodgy copy and 1 good copy, and same with dad. But because it's a recessively inherited disease, they're all good. They're just termed carriers. Now, what percentage of, so if they were to have offspring, what's the possible genetic outcome of the offspring? Well, it can be big A, big A. It could be big A, little a. It could be big A, little a, and it could be little a, little a. Now what this means is you can have of 100%, 25%, remember 25 plus 25 plus 25 plus 25 is 100. So 25% can be homozygous recessive. 25% are going to be homozygous dominant. But what we asked was about the carriers. So here, 25 plus 25 is 50%. 50% of the offspring are going to be heterozygous, aka carriers for the disease. So that's question 1. The next question asks if one parent is heterozygous with the dominantly inherited gene and the other parent is homozygous recessive for the good copy, what percentage of offspring will have the disorder? Let's take a look. So let's just say it's dad that has the dominantly inherited gene. That's the disorder. Now remember, if it's dominantly inherited, you only need one copy to get the disease, which means dad has the disease. Mum, in this case, is recessive, so homozygous recessive for the good version, which means the percentage of offspring that will have the disease will be big A, little a, little a, little a, big A, little a, little a, little a. So what does that mean? Well, 25% plus 25% is 50%. 50% of the offspring will have the disease. 50% of the offspring will have the disease. Now the next question is about X-linked inheritance, and I'm not going to give you the answer to this question, but what I'm going to do is I'm going to hopefully provide you with the ability to answer it yourself. So all I've spoken about inheritance so far with these Punnett squares have been the chromosome 1 to 22, what we call the somatic. But chromosome 23, that pair, is the chromosome that determines whether we are genetic male or genetic female. So for example, remember, XY means genetic male and XX means genetic female. So when dad and mum have offspring, what percentage of offspring are going to be male, what genetic males, what percentage are going to be genetic females? Well, XX, female, XY, male, XX, female, XY, male. 50% genetic male, 50% genetic female. Now have a think. Let's just tag this, let's tag the Y from dad like that. So that means there's the Y and there's the Y. So obviously, every single male gets their Y chromosome from their dad, which means let's tag this X here like that, and let's tag this X here like that. So we've got what do we see? We see that because this is mum, that X came from mum, that X came from mum. 100% of male offspring they'll always get their X chromosome from their mum because they always get their Y chromosome from their dad. So they're left with getting the X chromosome from their mum, which means if mum has an X chromosome linked disorder, it's likely the son is also going to have it. Now think about it like this. Let's now just say that this little thing on the arm here means it's a disease version. There's a disease version of the allele on that X chromosome. Now, luckily, mum's got another X chromosome to back it up. So mum doesn't manifest the disease, mum's just a carrier. Now, if we have a look and see where it goes, you'll see that 50% of the females will also get that diseased X chromosome, but they also have a good copy, so they don't get the disease. So mum will be a carrier, 50% of the females will also be a carrier, but none of them will have the disease. But for the males, 50% get that dodgy version of the X chromosome, but because there's no good version of an X chromosome here to make up for it, 50% of the males get the disease. 50% of the males get the disease. 50% of the females are carriers, but 50% of the males get the disease because there's no X chromosome to back it up. Now, hopefully, you can answer the final question.